Welcome back. Today we are diving deep into, well, frankly, one of the most powerful and ethically tangled stories in modern medicine. It's the incredible saga of the HeLa cell line and the woman, Henrietta Lacks, whose identity it carries.
hmm And our sources for this really bridge two worlds, don't they? You've got the hard science, the lab breakthroughs, and then you have the very personal, often painful history of a family caught up in something enormous for decades.
Right, so our mission today is to weave those together. We want to unpack the science of it. How one woman's cells literally became the cornerstone of modern medicine.
Absolutely. But also you grapple with the profound injustices, the lack of recognition her family faced for so long.
It really starts with this almost unbelievable biological HeLa cells. They were the first human cells ever grown in a lab that just didn't die. They were called Immortal.
Immortal, unlike everything else tried before, which just fizzled out quickly. These cells, they reproduce an entire generation every 24 hours.
And here's the bit that really gets you, the listener. Henrietta Lacks's cells have now been living and dividing outside her body for far, far longer than they ever lived inside it.
Yeah, it's staggering. They're essentially a global biological entity now.
So to really understand the ethical side, the human cost, we need to start with Henrietta herself.
We do. She was born Loretta Pleasant in 1920 in a little place called Clover, Virginia. Grew up working on a tobacco farm.
And this wasn't just any farmland, was it?
No, it had history. These were the same fields that her enslaved ancestors had worked. She worked them too. She only stayed in school until the 6th grade, which, wasn't uncommon then for black children of the rural South.
But then, like so many others, the family moved, part of the Great Migration.
Exactly. Henrietta and her husband, Day, they moved north to Turner Station, Maryland, near Baltimore.
And Turner Station was seen as a kind of beacon, right?
It was. People called it the Promised Land. The Sparrows Point steel mills nearby were booming, huge demand for workers. So it became this striving, tight-knit black community, people building lives.
Okay, so setting that scene is crucial. Because then we get to January 1951, her first visit to Johns Hopkins.
Right. She complained of a knot, she said, and pain. Now, going into Hopkins then, you have to picture it. It was heavily segregated.
Separate waiting rooms, bathrooms.
The whole colored system. And Hopkins, while it provided care for the poor, black and white, there was this underlying attitude, very common at the time. Doctors often saw patients in the public wards, especially poor black patients, as research material. Taking tissues without explicit consent, well, it was almost seen as payment for the free care.
That idea, the patient not as a person with rights, but as a source of material, that's really the root of the problem here.
It is. So she's diagnosed stage D epidermoid carcinoma of the cervix. And the treatment they gave her was actually quite advanced for the day. Terrifying, but advanced.
Those radium treatments.
Yeah, inserting thin glass tubes filled with radium right against the cervix. They were called BRAC plaques.
And there's a terrible irony there, too, isn't there? With the radium itself.
A grim one. The Hopkins doctor who actually invented those plaques and others who worked closely with radium. Many of them later died of cancers likely caused by that radiation exposure.
Wow. So they're risking their lives to treat her while simultaneously...
Well, simultaneously, her tissues being taken completely without her knowledge for research purposes.
Henrietta didn't survive long after that treatment started. She died October 4th, 1951, only 31 years old.
And the cancer had spread incredibly fast, much faster than the initial diagnosis suggested it should have.
Which was later explained, right?
Yes. Years later, looking back at the records, the diagnosis was corrected. It wasn't the more common epidermoid carcinoma. It was a particularly virulent adenocarcinoma, very aggressive.
And that unique aggression, that's key to what happened next in the lab.
Absolutely key. Because, see, scientists like George Gay and his wife Margaret Gay at Hopkins, they had been trying for decades to get human cells to survive in culture. And failing.
They just couldn't crack it?
Nope. The sources describe their efforts vividly. Using these complex, almost alchemical mixtures, witch's brews, they call them.
What was in them?
Oh, things like chicken blood plasma, ground-up calf fetuses, umbilical cord blood. Anything they thought might provide the right nutrients. They were desperate for a stable human cell line.
But every single attempt, every sample they tried...
Died. They all just died off quickly in the lab. Until Henrietta's.
So her tumor sample, taken during one of those radium treatments, again without consent.
Exactly. It lands in George Gay's lab. His technician, a woman named Mary Kubitschek, puts it in a culture dish. And this time, everything changed.
They didn't just survive.
They didn't just survive, they thrived. They grew like nothing anyone had ever seen. just kept dividing, doubling, consuming nutrients uncontrollably. It was astonishing.
A monumental scientific moment, but still tied to this human tragedy.
Deeply. And Mary Kubitschek, the technician, she had this moment of realization. She actually attended Henrietta's partial autopsy.
Why was there an autopsy?
Well, Day, Henrietta's husband, had only agreed to a partial one. She was told, vaguely, it might somehow help his children down the line.
Okay. And Kubitschek was there?
She was there, and she saw Henrietta's feet. And she noticed the bright red nail polish on her toenails. Chipped red polish. And it hit her. Like, really hit her. She said later, oh, geez, she's a real person?
Yeah.
This wasn't just abstract cells in a dish anymore. It was Henrietta Lacks.
That sudden human connection. Wow.
But that realization, powerful as it was for Kubitschek, was almost immediately swept aside by the sheer scientific need for these cells.
Right. The demand just exploded.
Instantly. Driven mainly by the polio epidemic. The National Foundation for Infantile Paralysis, the NFIP, they were pouring money into Jonas Salk's vaccine research.
And they needed a way to test it on a massive scale.
Billions and millions of tests. Using monkey cells, which they had been doing, was just way too expensive and slow. They needed something better.
And HeLa cells were perfect.
They were perfect. They were incredibly susceptible to poliovirus, which made them ideal for seeing if the vaccine worked. But the real game changer, the thing that made HeLa a global phenomenon, was something technical.
What was that?
It was their ability to grow in suspension. Meaning they didn't need to be stuck to the glass surface of a bottle or dish, which is how most cells grew. That limited how many you could grow, only as many as could fit on the surface.
Okay.
HeLa could grow floating in a liquid medium, constantly stirred. Think like a big vat. Suddenly, you weren't limited by surface area. You could grow astronomical quantities.
That's what turned it from a lab tool into an industrial product.
Precisely, a biological factory.
Yeah.
And soon enough, an actual HeLa factory was set up.
Where was that?
At the Tuskegee Institute, interestingly. Staffed primarily by black scientists and technicians. At its peak, it was churning out something like 6 trillion HeLa cells per week.
6 trillion. That's hard to even comprehend.
Isn't it? And from there, HeLa cells went everywhere, shipped all over the world. They became the absolute standard tool in labs.
Used for everything.
Everything. Developing drugs for herpes, leukemia, influenza, Parkinson's disease, mapping genes, understanding cancer. They even sent HeLa cells into space. Both the U.S. and the Soviets used them to test the effects of zero gravity.
Incredible reach, but this runaway success eventually led to a huge problem, didn't it?
It did. Fast forward to the 1970s, and scientists start running into trouble. It became known as the HeLa bomb.
The HeLa bomb. What happened?
Well, it turns out HeLa cells weren't just robust. They were aggressively robust, almost invasive in a lab setting. A geneticist named Stanley Gartler made this shocking discovery.
What did he find?
He found that HeLa cells had secretly contaminated dozens, maybe hundreds of other human cell lines that researchers thought they were studying all over the world.
Contaminated them, you mean taken over?
Essentially, yes. The HeLa cells outgrew and replaced the original cells in the cultures without anyone realizing. It meant that potentially millions of dollars, years of research based on those other cell lines, might be completely invalid. It was a crisis.
How did he know they were HeLa?
He used a specific genetic marker. Think of it like a rare genetic fingerprint called G6PDA. It's found almost exclusively in people of African descent. Gartler found this marker in all these supposedly different cell lines. It proved they all actually came from the same single source, a black woman. Henrietta. So to try and sort out this mess, this contamination issue, scientists realized they needed to go back to the original source. They needed genetic material from Henrietta's family to confirm the HeLa markers.
And this is when the family finally finds out, decades later?
Decades later. And they'd had absolutely no idea. None. Living in poverty, often without health insurance themselves, while their mother's cells were fueling a multi-billion-dollar industry.
So researchers contact them. How did that go?
Not well. Not well at all. The scientists needed blood samples from the children. They were looking for things called HeLa markers, more genetic identifiers, to help pinpoint HeLa cells.
Yeah, purely scientific reasons for them.
For them, yes. But imagine you're Day Lacks, Henrietta's widower. You get these calls from researchers at Johns Hopkins using complex scientific language.
Combined with his own accent and background.
Exactly. There was a massive communication breakdown. Day came away believing they wanted to test his children to see if they had somehow inherited that cancer that killed their mother.
Oh, no. That's terrifying.
Utterly terrifying for the family. And you have to remember the context here. This fear wasn't totally unfounded.
How
Well,
so?
there was a long, dark history in black communities, oral traditions about night doctors who supposedly kidnapped black people for medical experiment.
Right, those historical fears.
And on top of that, the news about the Tuskegee syphilis study, where black men were deliberately left untreated for decades, just so researchers could study the disease, had recently broken. It was huge news. So the Lacks family's fear and suspicion, it was rooted in real historical trauma.
And this whole situation must have been agonizing for the children, especially Deborah.
Oh, profoundly. Henrietta's daughter, Deborah Lacks. She suffered immensely. The stress and anxiety from these calls, the confusion, it caused her multiple near strokes. Her quest became almost desperate.
What was she looking for?
Initially, it wasn't about the science or money. It was about her mother. She barely remembered Henrietta. She'd ask researchers the simplest, most human questions. What did she smell like? Did she like to dance? What was her favorite color? Things nobody can tell her. She and her brothers also really struggled with the idea of their mother's cells being alive out there, being bought and sold. They feared injections, thinking maybe researchers were giving them bad blood related to the cancer.
It's just heartbreaking. The world knew her cells, but her own children didn't know her.
Exactly. And meanwhile, the commercial side was booming. George Gay had initially given the cells away freely to other researchers.
But someone started selling them.
Oh, yes. Distribution companies, like one called Microbiological Associates, which eventually grew into major biotech players. They started commercially producing and selling HeLa cells.
And made fortunes.
We're talking serious money.
Billions over the decades. Today, something like 17,000 patents involve work done using HeLa cells.
And the Lacks family saw none of that.
None of it. Living with serious financial struggles, lack of health care. It created this stark, terrible inequity. And it divided the family, too.
How so?
Well, some of the family members, like her sons Lawrence and Zachariah, felt strongly that they were owed financial compensation, given the immense profits generated.
That seems understandable.
Entirely. But Deborah, her focus remained different. For her, it was about recognition, about knowledge, about finally learning who Henrietta Lacks truly was.
Did they ever get closer to understanding, to connecting with the cells? There was a really pivotal moment much later. Deborah and her brother Zachariah were taken to a lab at Johns Hopkins.
Zachariah. He had a particularly difficult time with all this, didn't he?
He did. He was born just 4 months after Henrietta's cancer diagnosis. He had had a very troubled life filled with anger. He blamed the cells, blamed George Gay. He'd even said he hoped Gay would burn in hell.
Wow, so what happened at the lab?
A researcher showed them the HeLa cells under a microscope, showed them dividing, living,
Yeah.
immortality right there in a dish.
What was their reaction?
Deborah just gasped. She said, "Oh, God, oh, that's my mother." It was the closest she had ever felt to her, seeing this living part of her.
And Zachariah, the angry son.
It changed him. Seeing the cells, this tangible piece of the mother he never knew, something shifted. Decades of bitterness seemed to soften. He actually ended up thanking the scientist.
A moment of grace, finally.
A profound moment of connection and maybe some measure of peace after so much conflict.
So, when you step back and look at this whole story,
Yeah.
what's the takeaway? Where does it leave us?
Well, it's this incredible, almost unbelievable duality, isn't it? You have this unparalleled scientific gift. HeLa cells revolutionized biology, virology, genetics, cancer research, vaccine development. None would be where they are without HeLa.
The foundation of so much modern medicine.
Absolutely. But it came at this immense human cost. The betrayal of Henrietta, the decades of suffering and invisibility for her family.
And it really forced a conversation about tissue rights, didn't it?
It absolutely fueled that movement. Legally, there was the John Moore case around the same time, maybe a bit later, which established that generally, once tissue is removed from your body, you don't retain property rights over it.
Which is still largely the case legally.
Largely, yes. But the Lacks story brought the ethical and human dimensions crashing into view. The right to know, the right to be recognized, the right to consent. Their story resonated globally.
There was also that spiritual interpretation from her cousin.
Right, Gary Lacks. He offered this really powerful way of looking at it. He called HeLa Henrietta's spiritual body, referencing a passage in the New Testament, 1 Corinthians, about the mortal body being buried and the spiritual body being raised. Immortal. It captured that duality beautifully.
So today, things have changed somewhat. We have HIPAA, patient privacy laws. Publishing someone's medical records, like what happened to the Laxes, would be illegal now.
True. The privacy aspect is much stronger. But the core ethical questions around tissue research, They linger.
especially with all the samples already stored.
Exactly. We have vast biorepositories of human tissues collected over decades. The big question remains, does the collective good, the moral obligation to advance science and medicine, sometimes outweigh individual autonomy, especially if tissues are anonymized and used for non-commercial research?
Or should we be required to get specific, informed consent for every use of every stored sample, even if that dramatically slows down potentially life-saving discoveries?
That tension between individual rights and the common good, between acknowledging the person and advancing science that's the enduring legacy, Henrietta Lacks left us all to grapple with.
A profound question indeed. Thank you for joining us for this deep dive into the complex and crucial story of Henrietta Lacks and HeLa.